Hello ladies and gentlemen, you are tuned into Edu-ca-te. Today we're going to be doing a practice question paper based on protein synthesis. Remember that I already did a video on protein synthesis. You can watch it right on the channel. So today we're going to be practicing section B, which is your 2.1, which is protein synthesis. It has got 11 marks, so let us see if we can get all the 11 marks. So first of all, when you approach a question paper, you have to read first the instruction of the question. It says that the diagram below shows the process of protein synthesis. So it means that you have to think all about the process whereby proteins are made. So you can see that we have been given a diagram here. So before we answer the questions, we need to know how to interpret such a diagram. So in this regard, whenever we talk of protein synthesis, we know that there are two organs that are involved. This organ, which has got pores, is called the nucleus, whereas this organ is called the ribosome. Remember, we said there are two organelles that are involved in protein synthesis. So now let us analyze. We have been given a label here, X, and you can see that this label has got a double helix shape. So if they ask us what is molecule X, we know that this will be DNA. Why do we say that? We can see this shape. This is the double helix shape. Then we can see that DNA is going through a certain process. Then all of a sudden the double helix has unwinded, or it has changed its shape. So after unwinding, you can see that there's a certain process that happens that results in this other molecule, which is molecule S. So this other molecule, we can refer this to as messenger RNA, mRNA. So mRNA is this molecule. Why do we say it's mRNA? It is because we know that in the nucleus, when DNA unwinds, it creates mRNA. So here is our mRNA. There is a certain process that this mRNA was created through. So this process is called process V in the diagram. So what is process B? We only create mRNA through a process called transcription. So it's called transcription. Then you can see that here we've got nitrogenous bases, and this is single-stranded. So we are sure that S is mRNA. It's mRNA. So now we can see that this mRNA has moved out of the nucleus and went to this organ called the ribosome. We are sure that it's a ribosome because we know that it involves two organelles. Protein synthesis involves two organelles. So the process that is taking place here, we know that the process that takes place at the ribosome is called translation. So we know that certainly this process is translation. And then we can also analyze that this is the organ, which is the ribosome, whereby translation takes place. We can also see that this shape is actually a tRNA molecule because it is approaching the ribosome with these anticodons. And then you can see that here there are amino acids which are bonded together to form a required protein. So now let us answer the questions because we've analyzed the diagram. So going into the questions, 2.1.1 says identify the following. When they say identify, this keyword of identifying simply means that you have to state, or you have to just name, or you have to just look around for that particular thing and write it. You don't need to explain anything. You can see it's even 1 mark. So molecule X, we have said, is a DNA molecule. So we can write here and say this is DNA, because we saw that it has got a double helix shape and it's inside the nucleus. Then we have got organelle Y. Organelle Y, we have said it's a ribosome because there are only two organelles which are involved in the process of protein synthesis, and we are sure that this is a nucleus considering that there is DNA inside it, and transcription also takes place inside the nucleus. So this is the ribosome. Then now, so we have got 1 mark here, 1 mark here. Then now we go to 2.1.2. It says identify as well. So it means it's also going to be 1 mark. Once it says 2 marks on identify, it means that you need to identify two things. So now it says identify and it puts on 1 marks. So let us now identify. We've got 1. What is label 1? 1 is part of the anticodon. So we need to know what is 1 actually. So you can see that 1 pairs up with C. Let me take a highlighter. 1 pairs up with C on the mRNA. 1 and C, they are pairing up. So we only know that during this whole protein synthesis thing, the only thing that pairs up with C is G. So we can definitely write and say that our 1 is G, because they said we need to identify nitrogenous base 1. So the nitrogenous base that pairs up with 1 is C. So it means 1 has to be G, because we know that A pairs up with U in RNA as well as G pairing up with C. So now we go to nitrogenous base number 3. Nitrogenous base number 3 here, we can confidently see that it is pairing up with A on the mRNA. So what pairs up with A, we know that it definitely has to be uracil. So it means that we can write here that 3 is actually U, which is uracil. And then now they say that describe the role of DNA during transcription. So now we have to describe. Once we say describe, it means that now the marks are going to be more than just mentioning or stating something. So you have to describe what does DNA do during transcription. So let us look at DNA. Remember that during transcription, the DNA double helix unwinds. So you can write that as first point. I'm not sure what the memo says, but then you have to write that the DNA double helix unwinds. So this point is actually explaining the role of DNA during transcription. Remember, transcription is the process whereby DNA forms mRNA. So how does that happen? That is the role of DNA during transcription. So the DNA double helix structure has to unwind, and also what else happens? The weak hydrogen bonds, the weak hydrogen bonds between nitrogenous bases will have to break. That's another point we can mention. It is still DNA that is doing all these things. So we are actually being asked what is the role of DNA during the transcription process. So when the weak hydrogen bonds break, what actually happens? We know that the two strands separate, but then in this case we have to describe how does it do transcription. We can now say that one DNA strand serves as a template. One DNA strand serves as a template to form mRNA, because that's the main aim of transcription. It is to form mRNA. I think here you can get your full 3 marks. I'm still yet to check the memo, but then you have mentioned the role that DNA plays. How does it do that? First, it has to unwind. The weak hydrogen bonds have to break, as well as one of the DNA strand, after breaking of the weak hydrogen bonds, one of them will serve as a template to form mRNA. So I'm not sure what the memo says, but then we can move on to 2.1.4. So 2.1.4 here says, describe the part of protein synthesis shown as process W which occurs at organelle Y. So where is process W? Process W here we have said that it is translation. Remember that here we have to describe. When we describe something, it means that we are giving a real details on it as to exactly how does something happen. So now we want to describe what part of protein synthesis which is known as translation. So we have to take the points about translation. So when we answer a question like this on translation, we can actually, let me do this and write translation. So this is translation. So first of all, whenever they say describe a process and they do not name that process, you have to first name this process. We call this a compulsory mark. Sometimes they'll give you a mark for mentioning translation, but then if you do not mention it, since it has not been given in the question, they say describe the part of protein synthesis. You can see that it's a part of protein synthesis, yet they did not mention that it is translation. So when they say describe it, you have to first start by naming it. So when you describe translation, you have to mention a few points that tRNA has a specific, okay, let's just say tRNA carries specific amino acids. Then you have to also mention that when the anticodon on the tRNA, when the anticodon on tRNA matches the codon on mRNA, matches the codon on mRNA, then tRNA will bring the required amino acid. Then you can continue and say that these amino acids will be bonded, which will be bonded by a peptide bond, which will be attached or bonded by a peptide bond to form a required protein, So in this case, you have described translation, but then in the question they did not tell you what is translation. They did not tell you to actually describe translation. They just said the part of protein synthesis. So when they hide what you have to describe, first start by writing translation first, as I've written here. I'm still going to do more practice question papers with you guys. Thank you for watching.